Hi guys, so we have Igcse paper biology 2024 May June paper 4-3. We'll go through the paper and see what it needed. Alright, so let's start. Question number 1 says figure 1.1 is a diagram of an animal cell. As we can see it over here, it has different parts labeled A, B, C and D. Now let's see the questions. The questions say here, complete table 1.1 by writing the name and function of the labeled parts that have been shown. Okay, all right, so let's start with the part labeled A. The part labeled A here is the nucleus. All right, and the nucleus is a part of the cell that stores genetic information or hereditary information. So part A is the nucleus, and what it does is that it stores genetic information or genetic materials. All right, yeah, that is what it does. Then the part labeled B. The part labeled B here is the cytoplasm of the cell. And cytoplasm of the cell, this is a part in which chemical reaction take place. So B is cytoplasm, and in cytoplasm we say that this is a site for chemical reactions. Okay, yeah, then we move to look at part C. Part C, this is the cell membrane of the cell. All right, and the cell membrane of the cell, this is what controls what gets into or out of the cell. So for C, we will say that this is the cell membrane. All right, and what does it do? We say that it controls entry into or out of the cell. This is what the cell membrane does. Then we have part D. Part D is the mitochondria. All right, and mitochondria, this is the site for aerobic respiration. This is where energy that is needed by the cell is produced. So we'll say D is mitochondria, and the function for mitochondria is aerobic respiration. All right, yeah, so this is the case. Then we move further. Explain how water moves into and out of animal cells. All right, so water moves into and out of the cells by osmosis. We'll state it by osmosis. But then again, we have to explain what osmosis is. We say that osmosis is the movement of water molecules. Definitely, water in biology, we speak that it moves by the process of osmosis from high water potential. Okay, this water has to move from a region where there's high concentration of water. But where does it go? It goes to lower water potential. All right, but also it has to pass through something. What is that thing? This thing is a partially permeable membrane. So when we are talking of osmosis, we understand that it's the movement of water molecules from a region of high water concentration or water potential to a region of low water potential through a partially permeable membrane. Roman 2 says a sample of red blood cells was taken from a person. The red blood cells were put into 3 test tubes. Each test tube contained a different liquid. All right, we had a test tube that had in it blood plasma, we had a test tube that had in it pure water, and we had a test tube that had in it a very concentrated solution. All right, all these were present. Now, figure 1.2 shows the appearance of the red blood cells when examined under a microscope. Identify the liquids that the red blood cells were immersed in. Write your answer. So, the appearance of the red blood cell should tell us in which liquid it was. For example, the one that in red plasma should retain the shape of the red blood cells. But the one that is in pure water, it means it's in a region with high water potential. Thus, water will move from the container into the red blood cells. We expect it to burst at the end of the day. But the one in a very concentrated solution, it means water will be moving from the red blood cells into the concentrated solution. So this cell will eventually shrink. Now coming back to the diagrams, when we look this first diagram here, the red blood cells are in the original shape, the disc shape. So definitely this is the blood plasma. And when we look at the second diagram over here, we see that they are kind of shrinking. They do not appear as this as they are here. So this is going to be a very concentrated salt solution. A very concentrated salt solution. Then after that we have this one here. This one here is a cell that has appeared to burst. So this is the one that was placed in pure water. Okay, then we go further. State the name of the molecules in red blood cells that combine with oxygen. Now we know that red blood cells usually transport oxygen, but they have an adaptation to them that enables them to bind to what? To the oxygen. And this thing is usually known as hemoglobin. So hemoglobin is present in red blood cells and it binds with oxygen, enabling the red blood cells to transport the oxygen. Okay, figure 1.3 shows a drawing of a white blood cell. Identify the type of white blood cell that has been shown. Now, white blood cells are of two types. We do have the phagocytes and lymphocytes. The difference between the two is that a phagocyte has a lobed-like nucleus, a lobed-shaped nucleus. But the lymphocyte usually has a large nucleus that is almost filling the whole cell. Now, from this explanation, you can definitely tell that this is a phagocyte. So we'll say this is a phagocyte. All right, then we go further into question number 2. It says here, figure 2.1 is a photograph of a spider plant known as Chorophytum comosum. As spider plants can reproduce by producing an identical plantlet, which grows away from a single parent plant. Now we can see we have a parent plant over here and we have a plantlet growing from the parent plant. Now the question says identify one feature in 2.1 that shows spider plants are monocotyledons. We have features of monocotyledons. One of them is that it has long leaves, long narrow leaves, but we also see parallel veins in the leaves. Now when we look at this diagram over here, we can actually tell that it has long narrow leaves. So one of the features identified over there or seen over there is long narrow leaves. Okay, then the next question says describe the advantages and disadvantages of a sexual reproduction in a population of spider plants in the wild. Now we have to know that a sexual reproduction is a kind of reproduction that does not involve fusion of gametes. Apart from not involving fusion of gametes, it usually involves only one parent. All right, now because it involves only one parent, we do have the advantages of only having one parent in this kind of reproduction. One of them is that it is fast because it's just occurring within the same plant. All right, but then also apart from that, we say that it does not require pollinators. All right, because the pollen do not need to be transported from here to there. All right, but then again, another advantage is that it gives offspring identical to the parent. Now if you want offspring that look exactly like the parents, we say that a sexual reproduction is very much befitted for it. So here we say offsprings are identical to parents. They look exactly like their parents. But then again, we have disadvantages. All right, apart from every advantage that we see, we do have disadvantages from this kind of reproduction. All right, one of them is that no genetic diversity. And why do we say that no genetic diversity? It is because the offsprings look exactly like their parents. So there's no a genetic difference between the two. But then again, apart from that, we say that they are more susceptible to diseases. All right, so when a condition like diseases attack this kind of plants, most of them will be affected. So we say they are susceptible to diseases. Okay. Yes, and also because they look identical to each other, we expect them to have high rate of competition. All right, so they'll compete for the same kind of what? Of resources. Okay, yes, so those are some of the disadvantages of what? of having a sexual reproduction. Okay, then figure 2.2 shows pollen that has been released from the flowers of hazel tree, Corylus avellana. These flowers are wind pollinated. Now we can see just from the diagram, you see the anther is releasing a lot of pollen to prove that this one is wind pollinated. And then explain why hazel tree has to produce large amount of pollen. All right, so the pollinator here is wind. Okay, so wind is expected to carry the pollen from maybe the anther to the stigma of the same tree or of any other different tree. Now, because wind is just blowing, it has to make the kind of pollination reliable. It means a huge number of pollen should be produced. So at the end of the day, we say that to increase the chances of successful pollination. All right, to increase chances of successful pollination. All right, so when I say successful pollination, I mean that successful transfer of the anthers of the pollen from the anthers to the stigma. That is the point. All right, then describe how a stigma of a wind-pollinated flower is adopted for pollination. Now, because it's only depending on wind, it means the stigma also has to have features that can easily trap pollen that is carried in wind. All right, so what are these features? You find that the stigma also is large. You find the stigmas are just hanging outside so that if wind has pollen, it can easily trap them. But also you find that the stigma is feathery. So we can say that they have large feathery, right? Yeah, so this is a feature we can see on stigma. And then the hazel tree uses cross-pollination rather than self-pollination. Describe cross-pollination. Now, cross-pollination, this is the transfer of pollen from the anther to the stigma of a different plant, but of the same kind. So we can assume, let's say we have plant A over here. Let's label this A, and then we have plant B over here. But there is a transfer of pollen from A to B. This transfer is what is going to be named as cross-pollination. But these plants have to be of the same kind. So we'll say that cross-pollination is the transfer of pollen from the anther to stigma of a different plant of the same species. They have to be of the same species so as it's a successful pollination. And then outline the events that occur to a plant after pollination. Now, what do we expect will occur to a plant after pollination? We need to understand that when pollination occurs, the pollen land on the stigma. All right, so after landing on the stigma, there is a growth of a pollen tube. The growth of the pollen tube occurs in the style. Mind you, the style connects the stigma to the ovary. All right, yeah. So there'll be growth of a style, and through this growth of a pollen tube through the style, and within this pollen tube, the pollen will travel. Okay, will travel from the stigma to the ovary, and when inside the ovary, the pollen will fuse with the ovary to form what? A zygote. So first of all, we'll say that after pollination, pollen land on the stigma. All right, pollen land on the stigma. Then a pollen tube grows in the style. In the style that connects stigma to ovary. That's the next thing. But after that, pollen travels through the pollen tube into the ovary. So it travels until it reaches the ovary. All right, then in the ovary, pollen fuses with ovule or egg to form a zygote. All right. So this is what is happening during the whole process after a pollen lands on the stigma. Then we move to the next question. It says here, proteins are biological molecules. Explain how proteins are made. Now, protein making usually occurs in two main steps. We have the first step, which is known as transcription, and this usually takes place inside the nucleus of what? Of the cell. It involves the formation of a messenger RNA. After that, we have translation. Translation occurs after transcription, and it involves the ribosomes assembling specific amino acids. But these amino acids have been brought to the ribosome by messenger RNA. So protein synthesis, we say it involves transcription and translation. It involves transcription and translation. Now let's start with transcription. We'll say here transcription is formation of messenger RNA from DNA of a cell. All right. So the messenger RNA is formed from DNA of the cell. All right. So it's just a copy of the DNA, but the actual DNA remains within the nucleus of the cell. Then we say the messenger RNA moves into cytoplasm of the cell. This is what is happening. Now, what is next? In cytoplasm, messenger RNA collects specific amino acids. All right, it collects specific amino acids and brings them to ribosome. Now, what does ribosome do? The ribosome assembles the amino acids to a protein. Okay, this is translation. So translation is when the messenger RNA brings specific amino acids to the ribosome, and the ribosome assembles them to give a specific protein. And those are the two processes involved in protein making. All right, so we go to the next question. It says here, figure 3.1 is a diagram of enzyme 1 catalyzing the breakdown of molecule A into smaller molecules B and C. Now we can see enzyme 1 here, it has binded to the molecule A and it breaks down into molecule B and C. Then after that, molecule D is shown in figure 3.2, which is here, and it is added into the mixture. This causes the rate of production of molecules B and C to decrease. Then the question says, using the information in 3.1 and 3.2, suggest why the rate of production of molecules B and C decreases after molecule D is added. Now we can go back. In order to tell why rate of production decreases, we have to look at the shape. Right? If you look at the shape of molecule A and the shape of molecule B, there is a similarity. Okay, so it means that both molecule A and molecule B can bind to the active site of the enzyme. Now, in case molecule D binds to the active site of the enzyme, it means molecule A will not be able to bind. And if molecule A is not able to bind, there will be no formation of smaller molecules B and C, which are definitely the products. Okay, so here we say that molecule A has a similar shape to molecule D has a similar shape to what? To molecule A. Okay, molecule D has similar shape to molecule A. Hence, it binds to active site of the enzyme. Active site of the enzyme. Okay, of the enzyme. Then after that, then no enzyme substrates are formed. It's no enzyme substrates are formed, and then less molecules, whereas less products are formed. Okay, so it's basically a competition between molecule A and molecule D. What are they competing for? They're competing for the active site. So in case the molecule A binds, we'll have the products. But when molecule D binds, we'll have no products formed, and then the rate of production will be lowered or will be decreased. Then Roman 2 says, figure 3.3 shows enzyme 1 after a change in its environmental conditions. Suggests 2 changes that could have caused the effect on enzyme 1. Now, if you look before, enzyme 1 had this kind of an active site. But then after some changes, it has this active site. Now, we do have some conditions that can cause change in active site of the enzyme. One of them is usually increased temperature. Okay, so when we have increase in temperature, we can result to change of the active site, because it's going to damage the bonds within the enzyme. But apart from temperature, we can also speak of increased pH. Increased pH also can damage the shape of the particular enzyme. All right, let's go to the next question. Okay, number 4 here says, explain. It says here, characteristics of plants are controlled by genes. Alleles are alternative form of a gene. Explain how new alleles are formed. Now, we have already said alleles are alternative form of a gene. Now, in case we are talking of how new alleles are going to be formed, definitely there has to be a mutation. Okay, so we'll say that they are formed through mutation, but we have to explain what is this mutation to the extent that it forms new allele. So we say mutation is a change. Mutation is a change in base sequence of DNA. So if we have a change in base sequence of a DNA, it means we can have a change in what? In the product that is formed. All right. B says the number of hours of daylight affects when plants produce flowers. This ensures that flowers are produced at an appropriate time of the year for each species. This response to the number of hours of daylight is controlled by genes. Wheat is a cropland. A farmer wants to produce a type of wheat that flowers when there is a fewer hours of daylight. Describe how the farmer could selectively breed this type of wheat. All right, so according to the question, the farmer wants a wheat plant that can produce flowers with fewer hours of daylight, rather than taking a long time before it could produce flowers. Now, how are they going to achieve this goal? First of all, the farmer has to actually select plants with desirable features, meaning that he has to select a plant with a trait of using fewer hours of daylight to form flowers. Okay, and then after selecting these plants, he will cross them together or breed them together. All right, then after breeding them, we will have offsprings, but then again we will choose offsprings that have acquired the desired character. And after choosing these offsprings, we breed them more. We breed them further. It means it's a repetition of breeding these characters that we want, to a point that we will have only the plants that we need. Okay, so first of all, what will the farmer do? We'll select plants that use fewer hours to flower. All right, then what will they do? We'll breed these plants. All right, we'll breed these plants, or we can say we'll cross these plants. All right, to produce to produce next generation or to produce offsprings. Then after producing offsprings, what will happen? We'll select offsprings with desirable features. Select offsprings with desirable features, the feature that we want. Okay, yes. After selecting them, what do? Then again, cross the offsprings. Cross the offsprings. All right, yeah. Cross the offsprings for many other generations. So it's something that the farmer will repeat until we eventually have a wheat plant that only takes few hours to flower. And then wheat is grown from seeds. State through conditions needed for germination of seeds. Now, conditions needed for germination are such as water, suitable temperature, and of course oxygen gas. Now, our question needs us only to state two, so we can state water, we can state oxygen also. All right, then we go further. Roman 3. Different types of wheat have also evolved through natural selection. Explain how natural selection differs from artificial selection. Now, first of all, let's understand natural selection. Natural selection, this is a type of selection that occurs gradually or slowly, and it is usually influenced by the environment. Okay, so changes in the environment result into what? A change in genetic makeup of an individual, and these changes, which are occurring slowly, are being transferred or transmitted to the next generations. Eventually, with time, we have individuals that look a bit different from what? From their ancestors. While with artificial selection, it's based on people's desires. So first of all, we know that natural selection, it is stimulated by the environment or pressured by the environment. So it is stimulated, or we can say pressured by the environment. That is the first thing. Okay, then after that, it occurs slowly. This is another thing of how natural selection is different from what? From artificial selection. All right, then again, I will say that suitable individuals are fit or selected for survival. All right, that's another feature, and they pass their traits to next generations. Okay, yes, and of course it affects many characteristics of this particular species that we are talking about. This is how natural selection is different from artificial selection. C. Scientists can selectively breed for increased pest resistance in wheat. Explain the benefits of growing wheat with increased pest resistance. So now we are growing wheat that does not depend on what? That is not affected by pests. So it's resistant to pests. One of the advantages of this one is that we'll have less use of pesticides. In short, less use of application of chemicals. So less use of pesticides. Okay, yeah, that's one advantage. And then another advantage is that we will have less pollution because we are not applying a lot of chemicals which are used as pesticides into the environment. So less pollution to the environment. All right, but also we'll have less spread of diseases that can result from use of these pesticides, but also will prevent death of or loss of pollinators, which would be affected by the use of pesticides. All right, yes, so those are some of the advantages of using pest resistant wheat. Okay, we are on to question 5. Phytoplankton are microscopic organisms that photosynthesize. State the chemical equation for photosynthesis. Now we know that photosynthesis is the process in which carbon dioxide combines with water to form glucose and what? And oxygen. Now we have to write chemical equation instead of words for this process. So it is carbon dioxide reacting with water. What do we get? We get glucose plus what? Plus oxygen, but we have to balance. And the best thing is that you can see we have 6 carbons here, so we have to ensure carbons are 6. We balance with the 6 here, but we have 12 water here, and we only have 2 hydrogen over here. So to get 2 hydrogen and 12 hydrogen over here, of course we balance by a 6. So C6CH2O will give us glucose, and of course we have 2 oxygen molecules over here, all right, while we have more than 2 somewhere else, all right. So of course we'll have to balance the oxygen also, and of course oxygen also balances by what? By a 6, all right. When you look at this question, you find that we have like here 12 oxygen plus this one here 6 oxygen, and of course this side here we'll have 12 oxygen plus 6 oxygen. When we add them together, we get the total number of oxygen, which will be 18. All right. A scientist B made notes about marine food web. Figure 5.1 shows the notes she made. In the notes, we see that in the sea, phytoplankton photosynthesize. Phytoplankton are eaten by smaller organisms in the sea called zooplanktons. Herringfish and codfish eat phytoplankton. Herringfish also eats zooplankton. Codfish eat other fish such as herring. Now, using the information in figure 5.1, we have to complete this food web over here. All right, but from the food web, it says that in the sea, phytoplankton photosynthesize. So they are definitely producers. They'll be here, phytoplankton. Right? And then, according to her notes, it says again, phytoplankton are eaten by smaller organisms in the sea called zooplankton. So we can see here that phytoplanktons are here, but then again they are eaten by smaller organisms. Now these are the arrows to show what can feed. We have krill, but we also have zooplankton. Yes. Then after that, she said that herringfish and codfish eat phytoplankton. So herringfish and codfish eat phytoplankton, but herringfish also eat what? Zooplankton. Okay? But herringfish also eat zooplankton. So herringfish should be here. Fish also eat zooplankton, and then here we will have codfish. All right? Here we will have codfish. Okay, then the next part says, hey, codfish eat other fish such as herring also. All right, yes. So the food web has been completed. Then we move further. State the principal source of energy for this food web. Now, the principal source of energy should be the thing that will give energy to phytoplankton, that will enable phytoplankton to photosynthesize, and this is primarily sunlight. Yeah. And then table 5.1 describes features of food web in 5.2. Complete 5.1 using information in 5.2. All right, so here we have the features and the maximum number in food web. Now we have trophic levels. We'll go back to the figure 5.2 to see the food webs. All right, so let's start here. If we take this path here, phytoplankton, this is 1 for first trophic level, right? We are looking for trophic levels, right? So phytoplankton in the first trophic level, zooplankton the third, herringfish second, herringfish the third, codfish the fourth, leopard seal the fifth, and Olca the sixth. And then you have again phytoplankton will be the first. When we go to Adele penguin, it will be the second. This will be the second if we take that path, and then Olca will be the third. And then if we take the path of phytoplankton, zooplankton, herringfish, codfish, that will give us 6. If we take the path of phytoplankton, herringfish should be the second, zooplankton, codfish the third, leopard seal the fourth, and Olca will be the fifth. And then we have phytoplankton, krill, crabby seal, and that will also give us 3. So we can see that the most of it has what? 6 trophic levels. So maximum number is 6. Then we go to primary consumers. Now we find primary consumers, these ones feed on producers. All right, so we can check here. We have the first one is zooplankton. it directly feeds on phytoplankton. And we have krill, it directly feeds on what? On phytoplankton. But we also have herringfish over here. We can definitely see that herringfish also is the third one, right? It feeds on zooplankton, phytoplankton. But we also have codfish. Codfish is the fourth primary consumer because it feeds on phytoplankton. But also we have adil penguin. It can also feed on phytoplankton. So we have 5 of them. All right. So we'll write here 5. And then tertiary consumers. All right. So tertiary consumers, these ones feed on secondary consumers. All right. So let's see. They feed on secondary consumers. So if this is a primary and then this is a secondary, codfish is one of the tertiary consumers. All right. Then if this is a primary, this is a primary, this is a secondary. All right. That's one. And then we have primary crabia, and then that will be again a secondary. But then again, when we take phytoplankton, codfish would be primary, leopard bear would be secondary, orca would be the second tertiary consumer. That's one path. But then again, if we take phytoplankton as primary, adil as primary consumer, leopard again is secondary consumer, orca will again be the third consumer. So we have orca, we have leopard seal, and we have codfish, which can fit into tertiary consumers. So we have a maximum of 3. All right. Then figure 5.3 is a photography of orca. Okay. Using the information in 5.2, predict and explain the most likely effect of a decrease in orca population on the size of krill. Okay. So when we have a decrease in orca here, orca, and how it will affect krill, this one here, krill. All right, so in that case, if we have a decrease in orca, we'll have a decrease in krill as well. Why? Because we will have a lot of predators that can feed on what? On krill together. So say that decrease in population of orca will result to decrease in of krill population due to presence of many predators. Okay, as these predators were ate by what? By orca. And then we have to also state the effect of decrease of orca population on the effect of what? On leopard cells. Okay, so here's leopard cells. Okay, so when we have a decrease in orca, we will have what? Increase in leopard cell. Why? Because we don't have a lot of organisms that can feed on what? On leopard cell. So we'll say that we'll have increase in leopard cell as they have no more organisms to eat them, as there's decrease in predators. So there'll be increase of leopard cells due to decrease in their predators. Okay, this is what is expected to happen. All right, then krill and seals can be harvested to produce omega-3 fatty acids. Many people take omega-3 fatty acids as a dietary supplement as they are thought to have many health benefits in human. List the chemical elements in all fats. Now, we do have elements that we find in fats that are usually carbon, hydrogen, and oxygen. And according to our question, we have to list them. So we'll list carbon, we'll list hydrogen, and we'll also list oxygen. Okay, using the information in figure 5.2, explain why it is more energy efficient to use krill as a source of omega-3 fatty acids rather than cells. Let's see their position in trophic levels. Okay, so krill is in the second trophic level. We are comparing it to what? To seal. Okay, krill is in second trophic level, but seal is in the third trophic level. All right. Okay, now it is best to use krill rather than seal because krills are in the second trophic levels, thus they have more energy in them as compared to seals. Seals will not have more energy in them because energy is lost in each trophic level.
All right, yeah, so that is the point over here. So we say that energy is lost in each trophic level due to activities or processes like we have movement, due to processes like movement. All right, yeah, we have processes like excretion. Then krill is in second trophic level. It has more energy than seal. All right, now due to positioning of what? Of the due to positioning of the trophic level, krill has more energy than seal, which is has more energy than seal, which is in third trophic level. Okay, so positioning of trophic levels favors the use of krill than seal, because krill will have more energy than what? Than seal. And then D says, describe how fats ingested by humans are digested and absorbed. All right, so fats are usually broken down in the process of physical digestion by what? By bile in the small intestine. So we say that fats are broken down into smaller pieces by bile, a process called emulsification. Okay, it occurs in small intestine, or you can be specific and say in duodenum. It increases this emulsification. It increases surface area for enzyme lipase to chemically digest fats into fatty acids and glycerol. Okay, into fat acids and glycerol. Then the glycerol and fat acids are absorbed by villi in the small intestine. Okay, then we say that the lacteal of the villi absorbs fat acids and glycerol, and eventually transports it to the body. So that is how fat acids are ingested, are broken down and absorbed. Okay, we go to the next question, which is question number 6. It says here, figure 6.1 shows boreal toad, Igcse boreus. This toad is listed as an endangered species. State the genus of the boreal toad. Now, the genus is definitely obtained from the biological name over here. This one here. This first name, which has started with capital letter, is the genus, and this second name here is the species name. So, Igcse is going to be the genus name for this one here. Yes, so this is the genus name. And then the second part says, one reason for boreal toad's recent decrease in population is a disease caused by chytrid fungus. State 2 reasons other than diseases that could cause boreal toad to become endangered. Okay, apart from diseases, any other thing that could affect these boreal toads in the environment could be pollution. So maybe exposure to different pollutants is also resulting to their decline in number. But also we can have hunting. Maybe they're being hunted for different studies. All right. Yes. So we could result into their decrease in number. Then C says scientists investigated whether increasing the number of beneficial bacteria that live on toad's skin could provide protection against fungus. The scientists took 42 toads and placed 21 in group 1 and 21 in group 2. Okay, so in group 1 we had 21 toads and in group 2 we had 21 toads. In group 1, the toad's skin was treated with beneficial bacteria. In group 2, the toad's skin was not treated with beneficial bacteria, so it's acting as a control group. The scientists measured the number of toads that became infected with fungus in both of them. Right, you can see, so they measured according to number of days and the number of toads that were affected with fungus every day. All right, so after time 0, none had fungus. After 20 days, for the both of them, 2 had fungus. And then after 40 days, for the one that were treated, only 3. For the one that were not treated, were 4. 60 days later, for the ones that were treated, were 9. For the ones that were not treated, were 17. You can see a difference over here. 80 days later, the number is also different, same as 100 days. And then calculate the percentage of 21 toads that have fungal infection after 100 days in group 1. The percentage of 21 toads that have fungal infection after 100 days in group 1. Now group 1 is here. This one here. This is the group 1 over here. Group 1. And of course, after 100 days, we only have 13 toads that have what? Have infection. So to get the percentage, it will definitely be 13 over the 21 toads that were what? Were infected. All right. So we divide here, and what we get, we multiply it to 100. Okay. Let me work it out and see what will be the answer. So when you work it out over here, you get approximately 61.9. Now we can approximate this one here to 62%. So this will be the answer. All right, we proceed with the next question. Using information in table 6.1, describe the effect of treating the toads with beneficial bacteria. Now, if we look at the table, when we treat the toads with beneficial bacteria, there is a less number of toads that is affected with what? With the fungal infection. You can see on day 40, we have only 3. After 100 days, we only have 13 that were infected. While when we do not treat them, all of them got what? Infected. So the benefit is that there is a less number of what? Of infection when treated with what? With infection, with bacteria. So we'll say here, bacterial treatments reduces chances of the toad being infected with fungus. All right. Yes, it reduces the chances of the toad being infected with fungus, and helping to conserve the toad population. All right, so this is what is happening. And then let's move further. This says there are some conservative programs to reintroduce boreal toads into the natural habitat. Describe why conservation programs are set for animals like boreal toad. Now, conservation programs are usually set for animals that are on the brink of extinction. Right, so that we do not lose them, we set these conservation programs. All right. Now, what are these? Why are these programs set? We are going to name them down over here. First of all, is to prevent the vulnerable species. Okay, so as to prevent what? Extinction. So we'll say here to protect the vulnerable species. Okay? This is one reason as to why they are being preserved, but also to protect the ecosystem. All right, to protect the ecosystem. Okay, and also we know that to prevent extinction of this particular species that we are talking about. Okay, but also we can speak of maintaining the food web. because these organisms are depending on one another. Okay, so maintaining food web or food chains is also important. And then we have this part here, Roman 2, which says conservation programs for other endangered animals such as birds use artificial insemination as part of captive breeding program. Describe how artificial insemination is carried out as part of captive breeding. Now we have to understand that artificial insemination involves a collection of male gametes from elsewhere and then introducing these male gametes into the female. Alright, now how do they do this? First of all, they extract or collect gametes from the male. So here we say collect male gametes from the male. Then after collecting them, what do? We screen the gametes. Okay, screen the gametes, or we can say like examine the gametes to check if they have any genetic problems and issues like that. Then after that, in checking the quality, then after that, what is done is that induce ovulation in females. Why do we induce ovulation in females? So that we can insert the gametes, okay, or insert the male gametes. And then after that, we say that insert the male gametes into uterus of females at appropriate ovulation time. At the appropriate ovulation time. At the appropriate ovulation time. Alright, that's why we induce ovulation, so that we can actually insert the gametes in or during the appropriate time. Yeah, so that is how artificial insemination is done.